Hi, good afternoon, everyone. On behalf of the UC San Diego Prepare Institute seminar series, I'm Dr. Constance Benson from the Division of Infectious Diseases and Global Public Health, and I will be co-chairing or moderating the session today. This is our inaugural seminar for the Prepare Institute. And for those of you who aren't familiar with our institute as of yet, it's a brand new effort undertaken by Dr. Davy Smith. The Prepare Institute refers to the Institute for Pandemic Response to Emerging Pathogens, Antimicrobial Resistance, and Equity. And Davy is particularly good at making up these acronyms, so we thank him for that. So our inaugural, if I could have the first slide today, our inaugural speaker for our first seminar in this series is Dr. Gene Tan. He's an assistant professor in infectious diseases at the J. Craig Venter Institute and an adjunct assistant professor in our Division of Infectious Diseases and Global Public Health here at UCSD. He will be talking on defining correlates of protection and markers of disease progression during SARS-CoV-2 infection. Just a few words about Dr. Tan, our featured speaker. He is a molecular virologist, largely focused on understanding the mechanisms of host immune system response against viral infection and disease. He received his PhD degree from Thomas Jefferson University, studying the molecular pathogenesis of rabies virus. He later trained as a postdoctoral fellow at the Icahn School of Medicine at Mount Sinai in New York, where he contributed to the design of candidate universal influenza vaccines and in the characterization of broadly neutralizing antibodies against influenza viruses. He joined us here in San Diego at the J. Craig Venter Institute as an assistant professor and was the director of the virus sequencing program. So I'll turn this program now over to Dr. Tan, and thank you for attending our seminar. Dr. Tan.
Thank you, Dr. Benson, for that nice intro. So today, actually I should
to have you start sharing your screen and stop sharing our host screen. There we go.
Right, so sorry about this. How do I share my screen?
So still, I think you might need to give him permission to share his screen.
No,
okay, got it.
Yeah, that was my fault.
Thank you.
Does everyone see my screen?
Yes.
My talk
Very
screen?
clear.
Yeah.
Great. Okay, so I've divided my talk, as Dr. Benson had mentioned, is defining the markers of disease progression and correlates protection during SARS-CoV-2 infection. So I've divided my talk into two main parts. And the first part is a collaboration with a friend colleague up in Stanford University with Tai Wang. And it's more of a mechanistic study on the antibody response following mild COVID infection. And in the latter half of the talk, I'll be discussing our effort in understanding the magnitude and the breadth of the antibody response against the variants of concerns, specifically against Omicron in comparison to wild type, Delta, and VOCs. So it's more of a very direct, straight serological analysis. So I wanted to, let's see, sorry, let me see if I can. Sorry, can everyone see my pointer? Yes.
Yep, I can see
Okay.
it.
All right, so I wanted to start with this slide since I think a lot of us work at this interface. We know that the interaction between pathogen and host is a very complex and dynamic relationship that's influenced by a multitude of factors. And I tried to include a lot of factors here. I know I'm not being exhaustive, but I feel like these are more than some important factors that we've studied in the past. But the end goal is to have an appropriate immune response that would either provide serum immunity or a timely viral, in this case, timely viral clearance with little or no symptoms of disease. So today I'll be mostly talking or focusing on our work in better understanding the role of the antibody response against SARS-CoV-2 in disease and in protection. As we all know that while our immune system is designed to protect us from disease, oftentimes that same response, if dysregulated, can also cause disease or harm us. So this is true for the role of B cells and the antibody response, but it can also be seen in other arms of or other compartments of the immune system, such as T cells, myeloids, derived cells such as neutrophils, or macrophages. And there's other factors that we can really delve into, but today I'll just mostly focus on the antibody response. So I'm not really going to talk too much about the virus itself because I'm pretty sure a lot of people are really up to date to some extent. So I'm just going to focus on a very, very brief intro to like the disease. So this is what the hypothetical lung tissue interface and the lower respiratory tract of a host in normal state, what you see in the left, and on the right side is when you would see infection with SARS-CoV-2. So hypothetically, or what you would see is that you would have SARS-CoV-2 directly infecting these lower respiratory endothelial cells and essentially triggering a cascade of inflammatory responses that would trigger vascular leakage, coagulation, and inflammation. And that could lead to the disease itself. But as most of us know, that the ACE2, which is the host cell receptor for SARS-CoV-2, is not only expressed on pulmonary cells, but are also found in other organ systems, such as the renal system, hepatic, GI, cardiac, and therefore this is where we see a lot of different manifestations in the disease itself, and it's actually outside a lung pathology that you're actually seeing. And just as a plug for my other colleagues here, if you're really interested in looking at cardiac implications of SARS-CoV-2 cardiac function, so you can, I have a colleague here named Norberto who is willing, more than happy to talk with you guys as well. So why study antibodies? Mostly because we know the antibody response against pathogens, and here to say specifically SARS-CoV-2, play a large and important role in immunity. And as most of us know, antibodies are this Y-shaped molecule, and you have two Fab regions here on identical Fab region that is involved in determining the specificity of the antibody and recognize a specific epitope. And that binding usually is critical for its specific attributes. So in a lot of ways, what you have binding will be correlated to what function it is subsequent to. So in this case, could be a neutralizing antibody or a non-neutralizing antibody. But what's often forgotten or really put aside is sometimes the role of Fc, which is the base of the antibody molecule. And as you will later see in my presentation, you'll hopefully better understand or better appreciate the role of the Fc, because since it has some, I'll say, structural determinants that would modify or modulate the function of the antibody in context with the other compartments of the immune system. So the Fc binds to its receptors called Fc gamma receptors and binds to C1q component of the complement. And as a result of this Fc to Fc gamma receptor engagement, you have the activation of cell-mediated immunity. Now, depending on what type of cell that Fc gamma receptor is expressed on, you would have subsequent or associated cell-mediated response following the engagement of the antibody to the Fc gamma receptor. So in context of a neutrophil, you could have the release of chemoattractants. you have release of cytotoxic substances, and you have phagocytosis. In context of monocyte and macrophage, you would have the oxidative burst. You have phagocytosis as well, similar to a neutrophil. So as you can see, that it also depends on how the antibody recognizes what in the pathogen in what context. So you could have immune complexes where you have a complex of the particle itself opsonized by the antibody, and that immune complex will trigger some sort of response in the subsequent in that respective immune cells. Or if in the other context where you have a virus infected cell, where the antigen is expressed on cell surface, and then you have antibodies binding to it, and then you have either a neutrophil or a macrophage recognizing that interface between the virus infected cell and your antibody, and there you have the induction or the activation of phagocytosis, and therefore the clearance of the virus. So how is Fc-mediated immunity regulated? So modulation of the Fc-mediated is essentially a role of the Fc, and in this case, we can largely say that it's due to two main determinants. One is the IgG subclass, and the second is composition of carbohydrates or glycosylation in the Fc region, specifically on the CH2 region of the antibody molecule. So as most of us know, there are 4 different subclasses of IgGs in humans, and depending on what IgG it is, it has different affinities to different Fc gamma receptors. And these Fc gamma receptors, as you can see here, can be subdivided into activating or inhibitory Fc gamma receptors. So green is activating, and the sole red Fc gamma receptor is inhibitory. So you have different subclasses of your IgGs that have different affinities for these different types of Fc gamma receptors. And as you can see that even these Fc gamma receptors are differentially expressed on different types of immune cells. And that really basically gives our immune system a lot of breadth and a lot of flexibilities in targeting or our immune system to the different pathogens. And depending on the innate immune cells that's involved, then you have different mechanisms or Fc-mediated immunity that is activated. So interestingly enough, if you were to go back here, I had mentioned previously that Fc gamma receptors are generally broken down into 2 different types. One's activating, one's inhibitory. Interestingly enough, activating complexes, activating Fc gamma receptors, generally speaking, are considered low affinity, meaning that they will have low affinity to Fcs of antibodies unless you have immune complexes. So you have avidity that plays into part in activating these low affinity activating Fc gamma receptors. And you see that here on the left side, and in context of inhibitory, they're considered high affinity. So you really don't need that much to inhibit the activation of these Fc gamma receptors. And therefore, you see a lot of these, you see a lot of broad spectrum ways of modulating the response following Fc gamma receptor engagement. Now, the second part on how Fcs modulate Fc-mediated immunity is the glycosylation pattern on the Fc. So here you see a different representation of your Y-shaped molecule. This is your Fab of the antibody, and here is your Fc of your antibody. And CH2 specifically at site 297 is where you have modulation or modulation of your Fc by different carbohydrate groups. So specifically, I'll be talking about the citylation or sialic acid motif that is can be modulating the core Fc of any given antibody. And at the same time, you have the fucose here represented by the red triangle. So what you see here is that what we know is that Fc glycosylation impacts or influences the way they also bind or to Fc gamma receptors. So in what we've known in the past is that if you have high sial, let's say if you have no fucose, so if you delete this fucose, then you have high affinity to type 1 gamma receptors. And these are the type of type 1 gamma receptors I just showed you earlier in the previous slides, where you have activation of pro-inflammatory responses. So these are ones that are associated with phagocytosis, release of cytotoxic molecules. So therefore, they're a little bit more highly inflammatory. The second type of carbohydrate or glycosylation pattern difference is when you have high sialylation rate. So if you're going here, it's that you have sialic acid motifs, and that decreases the affinity to type 1, but increases the affinity to type 2. And these type 2 are CD23 DC signs. That is tended to be more activation of anti-inflammatories. So putting this all together, you can see a spectrum of different situations or in context of how the Fc gamma receptors are modulating or regulating the response to any type of infection. So on the left, you would have, if it's highly fucosylated with sialic acid motifs with low IgG1, it tends, generally speaking, tends to be more anti-inflammatory and more inhibitory. And that's defined by how the Fc engages Fc gamma receptor type 1 or type 2. On the other side of the spectrum, you have afucosylation, where as I mentioned earlier, no fucose tends to have high affinity Fc gamma receptor type 1, which is more associated with different types of pro-inflammatory responses. So what we are interested is looking at, are there antibody-based metrics that can predict the course of disease? Partly due to the fact that taking antibodies from sera is it's routine practice. And this is borrowed from work that Taya had done in the past with dengue, where she saw that as some of you might know, that secondary infection with a different type of dengue virus tends to increase susceptibility to increased disease. And that's associated with antibodies that are non-neutralizing and highly afucosylated, meaning there's no fucose on the Fc. And so we borrowed that observation, see whether or not we could see something in COVID patients. Now, what we were thinking about is that if we could find ways to predict the trajectory of people who would get more severe disease. And we thought a good way of doing that is to look for a cohort where they all presented with mild disease initially. So we found this cohort that were initially recruited to get lambda interferon. And we were specifically looking at just the placebo group. And what we saw here is that participants were enrolled within 3 days of a positive PCR test for COVID. And from there on, they were followed for almost a year. And what you can see here on the left is just the antibody neutralizing kinetics of these people from day of enrollment till month 10 post infection or post initial symptoms. And what we noticed here is that we saw a very small group of participants who progressed to more severe disease. And the way we define disease progression to more severe are either being admitted to the hospital or going to the ER. So what we noticed is that the majority of these people stayed mild, thankfully. But what we saw is that about 8 of these 120 patients were eventually, I think one died, and I think a couple were admitted to the hospital or went to the ER. And we saw this also in the second cohort. So we were interested in whether or not, whether there's something special or something unique about the antibody signature of these people who progressed to more severe from starting off from a mild initial progression. So the colors here that you see is basically the neutralizing antibody. So as you can see here is that the non-progressors, the NP here on the right side, had a variety of neutralizing antibody titers, either from very low or undetectable to very high. What's interesting is that progressors all had very little or non-detectable, not neutralizing antibody response at day of enrollment. So we looked at to see whether or not if there are differences in their fucosylation rate. So here on the left are this is binding antibodies, not neutralizing. So all of them have, in some way or form, have detectable antibody binding. But as when you compare it to the previous slide that had non-neutralized, they were not neutralizing. What's interesting is that both in the first cohort and the second cohort, their fucosylation rate was much lower, meaning, sorry for the different naming, but they were highly afucosylated, meaning that they had no fucose in the core FCs. And if you compare that to hospitalized patients, so this is a totally different cohort, we see that the people who progress had very similar FC afucosylation patterns compared to hospitalized patients. And so we tried to get into figuring out what, we were trying to dig deeper into the mechanistic aspect of why these were highly afucosylated. And alpha-1,6-fucosyl transferase is an enzyme that adds fucose to your FCs. And so we looked at and we isolated CD19 or plasmablasts. And as you can see here, that non-progressors have higher expression of FUT8 or fucosyl transferase, meaning that they tended to add fucose to their FCs, while people who were progressing to more severe disease had less expression of FUT8, which translates to less fucose or more highly afucosylated FCs. So putting that all together, what we saw and what our preliminary work was showing us is that if you were to combine the neutralizing activity or the lack thereof neutralizing activity and the lack thereof of fucose in their FC, that it can be predictive of people who start off having mild COVID to progressing to more severe disease. Now, there are some limitations to this, is that the reason why we used two cohorts was that we were trying to, convince ourselves, obviously, but convince our reviewers as well that, we had what, eight patients, right, for cohort 1 and 6 patients in cohort 2. So, very small, a very, very small proportion of a larger population, but I think it's indicative of something that is quite interesting in terms of predicting value on just based off of the antibody response or the SC. So once we looked at that, we were interested in looking at how does it compare to people who have been vaccinated, right? So we know that patients who end up being hospitalized have high afucosylation patterns in the FCs. People who start off having mild COVID but progressing to more severe had increasing afucosylation. What does it look like for vaccination, people who have been vaccinated? So these are people who have or participants who have never been infected by SARS-CoV-2 but were naive at the time of vaccination with mRNA. I think here's the Pfizer vaccine. So This is just the kinetics of the antibody neutralizing response following post-dose 1, post-dose 2, and post 3 months after immunization. As you can see that there is a peak of after dose 2, and most of the antibody response, the subclass of IgG being responding to this is IgG1. So what's really interesting is that what we found was that even though that fucosylation rate was not much different between vaccinated and the outpatient. So this is the timeline for this would be this is day 28, and this is also day 20. So we tried to match it time-wise in terms of following infection and vaccination. But what's interesting is that sialylation is much higher in the vaccine group. And so this is, if you were to remember the slides in the front of the presentation, is that sialylation tends to be more regulatory, more inhibitory. It does not really have, I would say, pro-inflammatory signature compared to afucosylation. So there's a distinct difference between people who are participants who have been vaccinated and not naturally infected. So to provide some, I would say, mechanistic understanding of what we're doing, what we did was we isolated these antibodies from sera and then made immune complexes with just the spike protein. So F0 means these are antibodies are from afucosylated participants. This is fucosylated, and this is antibodies from vaccinated, and this is just the antigen only. So at that time, we didn't really have a real challenge model. We had to use, this is a non-lethal, I would say, inoculation of your immune complex and see whether or not you would see some inflammatory response following the inoculation of these immune complexes to our transgenic mice. And what's really cool about these transgenic mice is that express human Fc gamma receptors. So we are dealing with trying to recapitulate what we would see in humans and mice, since these have Fc gamma receptors from humans. So what we saw here is that if you were to inoculate these mice, these transgenic mice, with the afucosylated immune complexes, that you do have an recruitment of neutrophils into the bowel compartment of the mouse. And you also have increased infiltration of monocytes. And that's compared to the ones that have been fucosylated or the ones that were vaccinated. And this is our spike only control. And you see that there are, if you were to look at the bowel fluid, that you do see a lot of, so these are RNA-seq data. So these are expression of pro-inflammatory genes that are associated with the infiltration of these pro-inflammatory innate immune cells into the space where you were inoculating these mice with. So what's interesting is that if you were to, so what we had was we also had CD16 knockout mice. So if you were to instill these same immune complexes into mice that lack the CD16, so these are Fc gamma receptor 3A, that you lose that infiltration or you lose that pro-inflammatory response, which suggests to us that it's CD16 dependent, which is quite interesting because you also have CD32, that's another Fc gamma receptors, but it seems like it's CD16 dependent. Now, I don't think we have CD32 knockout mice. I have to ask Todd about it, but it would be interesting to see. But from what we've seen so far, it's CD16 dependent. So as a conclusion to this part of the talk, so we saw the induction of early neutralizing robust neutralizing antibody that are fucosylated, and that is a good prognosis for resolving infection in mild COVID. And we observed this in 2 different cohorts. FUT8, which is an enzyme that is associated with fucosylating the Fc, is expressed higher in the B cells of non-progressors. So these were found in the CD19 and CD19 and plasmablast compartment, which is associated with higher fucosylation group in pattern in the non-progressing part. Afucosylate immune responses, immune complexes can initiate an inflammatory response in a CD16 or Fc gamma 3A dependent manner. And the antibody response following mRNA vaccination demonstrates distinct glycoform compared to hospitalizing patients. So therefore, we really do have a lot of future directions to do. I mean, in terms of even our mouse model, it's it's not the ideal, but for what we had at that time, we thought that that was giving us to validate what we see in humans, but obviously it's better to have a challenge model in the future. There's other Fc-mediated response, immune, I would say, mechanisms that we would like to address as well. This is to highlight our work right now with Marcelo Freire's group here at JCVI. And really looking at, Marcelo is a oral immunologist, but he's also very interested in the IgA, the mucosal response. And he's also an expert in IgA and neutrophil inflammatory response. So in terms of our work right now, we're looking at whether or not we can extend our study with Taya to see whether or not you have induction of pathological inflammatory responses that might, cause pathology rather than clearance of the virus. And it's interesting to see that these, for example, fibrinogen is highly expressed even in convalescent patients, participant, even though it's been 2 to 3 months after they've been exposed to the virus. So you have a heightened inflammatory response that still needs to be addressed in terms of people who have resolved the infection yet still have these signatures in their blood. So in the next part of my talk, I'll just talk about our contribution to understanding the magnitude and the breadth of neutralizing antibodies to SARS-CoV-2 and its variants. And I think most of us remember when we found out during Thanksgiving, probably dinner, that Omicron was a problem. And so thankfully, this was done collaboratively through a lot of people, and I'll discuss our collaboration in the acknowledgements part, but it's something that is worthwhile talking about. So as most of you know, that Omicron has just this huge amount of point mutations that was found in RBD, rendering what they thought at that time pre-existing immunity to be very, very low, and even with the boost. So what we decided to do was to compare what at that time was the predominant circulating variant called Delta, as most of you know, and then one that was considered to be the one that was associated with decreased pre-existing immunity, which is the Beta. So we know from past reports that Beta tended to elicit or tended to escape against antibodies and also pre-existing immunity against vaccine-induced immunity. So we chose Beta, Delta, and of course, wild type in comparison to Omicron, see how that works out in terms of pre-existing immunity. So we use a pseudotype viral particle platform as a surrogate for wild type virus, as most of you know. SARS-CoV-2 is a BSL-3 level pathogen. We really can't work with it in BSL-2. So this is the next best thing. And I apologize in advance if you know this. I'm pretty sure a lot of you guys know this, but just as a way to get everyone on the same page, what we do to make these pseudoparticles is first to express or transiently express our S protein, SARS-CoV-S into 293 T cells, and then we basically infect these with a defective VSV GFP virus. And after its infection, what comes out is a VSV particle that's pseudotype or expresses the S protein of SARS-CoV-2. And using this system, we should be able to find out whether or not antibodies that recognize the spike protein on this particle can elicit neutralizing responses. So the way we do this is that we have a pseudotype right here. We use this to infect your cell type of choice. And if you had infection, then you would have the expression of your GFP from that's encoded by the gene in this particle. And therefore, we use a plate reader just to count cells expressing GFP. And that would be a measure of infection. And if you have no infection, then you obviously have no GFP expression. So we made use of this system. What we thought about was that to generate a multiplex neutralization assay, whereby we express you utilize two different viruses at the same time, expressing two different fluorophores. So in this case, what we did was we made two different viruses. For example, one would have the VSV expressing the beta S, but this one would be expressing the GFP. And in this case, we would have another VSV, but it's expressing the S from the delta one, but it would be expressing the RFP. So essentially what you can do is you can co-infect these two different viruses. And what you can see here, and I think you can see it, is that you have different expression of different cells. So meaning that if you see a green, that would mean that would be a beta VSV entering into that cell. And then if you have RFP, then that would be a virus representing the delta virus. So we ended up using temperate cells expressing, viral cells expressing the temperate serine protease and the human AC2 cells, because it provided the best read on highly susceptible to this virus. And so what we did was we basically used this to multiplex our assay, and we were able to do 4 different viruses in two runs. So that saves time and saves precious precious volume from the patient samples. So this is what it would look like. So you'd have the delta right here. This would just be your cell, your virus expressing the delta S, and this is our control. So, and this would be our beta. So this is just a virus also. So what you can see here, this is a setup of our neutralization assay, whereby you serially dilute your virus, your sera from least diluted to most diluted, and these were done in duplicates. So as you can see that you have here's essentially neutralization of your SARS-CoV-2 delta, and that can be, it's pretty, I have to say this, it's pretty accurate and pretty robust in terms of readout. So we wanted to evaluate our work, validate our work against using real samples, right? So these were from hospital workers that were vaccinated with the Pfizer mRNA. So this is the same cohort that you saw in the previous section of the talk. And so these were participants that were given 3 doses of the Pfizer vaccine. We had day 0, day 21 is the first boost. The third boost would be sometime between 242 or 324. So we decided to just look at day 28, which is post first boost. And then this is and then looked at the other ones that I would say 2 time points after the third boost. No, third immunization. Sorry, I'm getting confused right now. So this is the neutralizing antibody kinetics following 1 and 2 boosts of the Pfizer vaccine. As you can see here, this is post dose 1, and you have as you have the highest neutralizing activity against the D614, which is the original Wuhan SARS-CoV-2. And as you can see here is that Omicron specifically had much more lower I would say, spread or range of their antibody titer response. What's interesting is that post-boost, post-third immunization, you see that you do have a boost after the week 1 and week 3 against Omicron, but it's significantly lower than Delta, D614, and Beta. And you can see that much more clear here at in this graph. So this is just an extrapolation of the neutralizing data that you saw in the previous slide. So we see here is that Omicron is much lower, as we all know, even after the boost. And this is 3 weeks after the second boost, so the third immunization. So essentially what happens is that you have a waning of your response even against beta. So this is 1 week after the third immunization, and you have Delta, Beta going down, and then Omicron going down. So I like showing this last slide as well, because it shows that, you know, qualitatively what it looks like in terms of the curves themselves. You can see that the Omicron curves are much, much flatter and lower compared to the ones that were against the wild type Beta and Delta. So these were the raw data that we extrapolated the NT50s from for the previous slides. So we established here in conclusion, what we did was establish a multiplex neutralization assay that can test 2 viruses at the same time, saving time and sample material. In healthy doses that received 2 doses of the Pfizer vaccine, the response against wild type Delta were comparable, but had lower responses against Beta and Omicron. 1 week after the third boost, response to D614, Beta, and Delta were comparable, but the response to Omicron, while boosted, was significantly lower. And that's not surprising when compared to what others had published as well. What we want to do right now is to make more viruses. So we're planning on making a blue and a far red expressing VSV virus so that we can multiplex, so that we can establish instead of using 2 viruses, we can hopefully use 4 viruses at the same time, therefore increasing our multiplexing capabilities and also saving obviously a lot of time and saving time and sample material. So I want to end with this. I think most of us are, you know, in terms of the way the vaccines are made, it's solely single, based on a single antigen, obviously, but the most important one, which is the spike protein. That really raises the question whether or not, whether we could in future iterations of the vaccine increase the breadth of the response against SARS-CoV-2, not only through including other antigens, but also hopefully adding the different compartments, being able to activate different compartments of the immune system so that we don't have to always play catch up and always have to figuring out what is the impact of this so-and-so variant against pre-existing immunity, if you were to hopefully provide second, third, or even four levels of protection by increasing the breadth through antigen and the responses that would hopefully be activated during immunization. So I wanted to end our talk with this. So we were interested, so this is collaboration with Richard Sherman, and we were thinking about really understanding the landscape of the antibody response in terms of the polyclonal response. We know that the RBD, the receptor binding domain, and the N-terminal domain is the main antigenic targets of neutralizing antibodies, for example. But are there other components of the spike protein that are associated with protection? And so the idea here is that analogous to what we had done in the past in terms of understanding the basis of breadth of protection against influenza by looking at different subdominant but conserved regions of the HA or the hemagglutinin of influenza A virus, could we do something similar in terms of identifying other parts of the spike protein that is associated with neutralization? So the idea here is to look for different region features of the spike protein, such as obviously the amino terminal and the receptor binding domain, as you can see here on the first and the third features, but there are other regions of the spike that have been associated with some neutralizing capacity. So what we're trying to do is make chimeric proteins or chimeric SARS-CoV-2 spike, in which we would graph the regions, SARS-CoV-2 regions, into another SARS-like spike that we in the general public do not have as much immunity to, and see whether or not we could use these as tools to identify antibodies that are associated with protection or neutralization. So essentially, we chose SARS-CoV-1 and MERS-CoV. So the idea here is to graph a region from SARS-CoV-2 and then replace it into, or substitute it into a backbone such as SARS-CoV-1 here on top, and MERS-CoV-2 here on the bottom. So you see here is that you would have a chimeric protein that would have the RBD from the SARS-CoV-2 in the backbone of SARS-CoV-1. Now, we know that there are definitely some cross-reactive antibodies that would, or I would say, conserve regions between SARS-CoV and SARS-CoV-2 outside RBD. But I think what we can do is we can use some, we can run, I would say, the wild type SARS-CoV to figure out whether or not there are to, I would say, normalize the antibody neutralizing response if we do see any. So we've generated 12 of these, 10 of these so far for the CoV-1 and CoV-2 chimeric pseudoparticles. As you can see that they come in variable viral titers. And so the idea here is to use the cohorts that we have. So people who have been naturally infected or people who have been vaccinated, see whether there's a difference in terms of the neutralizing capacity of these, and see whether or not we could identify regions of SARS-CoV-2 that would be associated with neutralization. So this is just a really very, I would say, preliminary data against using the wild type SARS-CoV-2 compared to SARS-CoV-1 expressing the RBD from SARS-CoV-2. So as you can see that it correlates really well, and I guess it shouldn't be too surprising that when you do a neutralization assay that compares wild CoV-2, SARS-CoV-2 compared to this chimeric, that since RBD is the main target of neutralizing antibody responses, that there's a one-to-one, almost a one-to-one correlation between the NT50 of against this pseudoparticle compared to this. So the idea here is to look for any other, to perform a similar type of assay against the other different chimerics and see whether or not we still do see any neutralizing activity. I think the smart thing is that we would have to normalize our data, our data using SARS-CoV-1. So essentially you would have, instead of using just two, we would have the chimeric here, and then you would have the full SARS-CoV-1 and see whether there's normalized for any background neutralizing activity. So this is, I'm ending my talk right now. So here we've generated 10 out of the 12 chimerics. The majority of the chimeric pseudoparticles have titers around 10 to the 4 to 10 to the 6, except for the one that's HR1, which had a titer of around 10 to the 3rd. Temperus HAC2 cells appear to be most susceptible to infection by these pseudotypes. And preliminary data demonstrated suggests the RBD elicits the majority of the neutralizing antibodies following mRNA vaccination. So this is not too surprising. So onto our acknowledgements. So obviously, we like to, we like to brag about the people who do most of this work, and it's so what we've done in the past in the first part of our talk was really led by Tai Long and her group, and we did more of a supportive group effort in terms of trying to work with her in understanding the mechanisms by which FC-mediated immunity and glycoform pattern impacts protection and impacts the disease progression in mild COVID. And I can't say anything better about our collaboration. It's really good to have people who you are friends with, colleagues, and you trust and are very competent in what they do. And it's always very refreshing to have those type of people coming alongside you. Pras was the PI for I think one of the Lambda cohort. So that's the cohort that we worked with in terms of trying to understand, the mild COVID infection. Matt was the instigator, I would say, in trying to figure out whether or not we would be interested in working with him in terms of understanding the breadth and the magnitude of the immune against Omicron. So, he was the one who really came alongside, really asked whether or not we would be interested in working with him in terms of trying to figure out. And it came out to be a really good partnership. Can't say enough about the people who have been able to provide us with all the valuable resources. These people, Bali Upinder, Scott Boyd, Mark Davis, Catherine Bush, Prabhu, are all instrumental in providing us with the Pfizer cohort, being able to freely give us and giving us the flexibility and permission to use their data, their samples, as well as Stephanie and Ariana, Yarden, Mary, who submitted, gave us our data for our pregnancy cohort, which I did not show today, unfortunately, just due to timing issues. So lastly, but not obviously, is our group here at JCVI. I can't really say anything better. Sanjay is a master in synthetic genomics. Without him, we wouldn't have been able to really do the Omicron study, especially Yang has magic hands and was able to really initiate the assembly of the Omicron and all the other variants and ship it to us here, and where we continue the cloning. Terry Gelbert is an amazing person who had worked with me this past year and during the pandemic, and really was there in the trenches with me because, obviously have very limited hands, and I can't say, not only hard worker, but very smart, always catches me doing a lot of wrong things. And lastly is Benjamin. Benjamin is a very special, I say boy, but not really boy, as a man. He came to me as a undergrad in the midst of during the pandemic 2020, and didn't really know anything about viruses at that point. All he wanted to do was, a lot of work and thinking how much he has gone through this past 2 years. He's been amazing. He's a very grounded, intelligent, very hardworking, and without him, a lot of this work, especially the neutralization assays, I'm not sure how many neutralization assays he's done, wouldn't have been possible. Actually, even the Omicron, he was back in Pitzer College up in the LA area. After Terry Young and I were finished our cloning and we're making the particles, I asked Ben if he could come down and finish, help us with, and he was in the middle of his, middle of his finals. He graduated one semester early from college. So, and he is an aspiring MD PhD student, and anyone would be very, very lucky to have him. So yeah, I hope that some of you guys would watch out for him if he does end up being in your institute. So that's it.
Wow, I want to thank you for what was really a superb discussion today. We have a few moments left for questions. If people have specific questions, you can unmute your microphones and ask Dr. Tan directly. I might take the opportunity to ask you one question. In any of your cohorts, particularly the ones where you looked at antibody responses to Omicron variants, have you had any opportunity to include in your cohorts either immunocompromised patients or people who've gotten a fourth booster dose of the Pfizer vaccine to look at longer term effects of those?
Unfortunately, no, not yet. We have reached out to the Kawasaki group at UCSD and looking at patients with, I guess, as immune compromised, I think, and so hopefully that would yield some interesting results. But in terms of like Kawasaki, where they do get a lot of IVIGs as a therapeutic, and that's really based off of like our limited understanding of how glycosylation impacts minimizing the severity of the inflammatory response, right? So,
Sure.
yeah.
Great. And, obviously the antibody response is not the only response that determines disease progression and outcome. Have you had an opportunity to collaborate with folks who are looking at the T cell response?
Not yet. I would love to, but obviously, I just saw a paper on CD8 T cells in terms of, I guess, the pathological effect of CD6. I'm not sure if it's. I think it might be controversial or conflicting, where CD8 T cells express CD16. That high expression CD16 tend to, obviously build into like whether or not it might increase the inflammatory response as well. So I'm not too familiar with the T cell response, but yeah, that'd be great.
Great. Well, that's obviously an area of strength here in La Jolla, and I'm sure some of our colleagues are on the Zoom call and may be interested in reaching out to you to kind of look at opportunities to collaborate in that area with the assays and work that you're doing.
Oh yeah, definitely. Yeah, that'd be really cool.
Great. Are there any additional questions from anyone in the audience that we can get to in our last minute here?
So, Gene, this is John Glass from JCBI.
John.
Your data suggest that to me that vaccination will result in a antibody population that can compensate for the afucosylated response that some patients get, but that is that true? Can vaccination overcome the effect of afucosylation?
I'm not really sure. I mean, it has not been really addressed in terms of, not fully. I know there's some preprints in trying to define the glycosylation patterns of people who have been infected and vaccinated, and people who have been antigenically, I would say, naive and then vaccinated. But we have not done that particular work yet. And I'm not really sure what would be the impact of people who have these higher afucosylation rate and then getting vaccinated, and what the IgG response would be following that the immunization.
And is there a human genotype associated with afucosylation?
You're talking about, you could say, then you're talking about maybe FUT8 variation, FUT8 expression. I'm not really too particularly sure if there's any FUT8 deficiency in the human population, if that's what you're asking.
But at least this suggests to me that pre-infection is definitely not as good as vaccination.
Pre-infection is not as,
Or COVID infection is not going to be as protective as vaccination.
yeah, I mean, there's a lot of multiple factors that goes into that in terms of, like, I guess the genetic variation in terms of how that person or individual responds longitudinally following a mild or even a severe COVID disease, and how they would respond to immunization. I'm not sure if I'm answering the question.
Thank you.
Great. Well, I think we're about out of time, so I just want to remind the audience that our next Zoom webinar in this series will take place on March 22nd from 4 to 5 p.m. And you'll be receiving further follow-up with details about that. I want to thank everyone for attending, and especially thank the speaker for what was really a very elegant body of work and a wonderful presentation. So thanks, everyone.
Thanks.